So before you begin your experiment, go ahead and turn on the UV light for at least 30 minutes to ensure that the biosafety cabinet is completely sterile. Also, before you open up the hash, ensure that the blower is on. And before you put all your equipment in that you'll need for the experiment, go ahead and spray it down with 70% ethanol. So what I'm doing in this experiment is I will be adding cold DMEMF12 into a 50 ml conical tube. The DMEMF12 has to be cold because you're going to be mixing it with Matrigel, and Matrigel solidifies when it reaches room temperature. The Matrigel is going to be retrieved from the minus 20 degrees Celsius freezer, and it's going to be mixed with the DMEMF12. DMEMF12 is a basal media that we use for that is used for culturing hiPSCs. So in my hand is the Matrigel. The Matrigel is at a concentration of 1 milligram per milliliter. What I'm going to do is I'm going to add 1 ml of the DMEMF12 straight into the microcentrifuge tube. Now what I'm going to do, I'm going to go ahead and mix the contents in the microcentrifuge tube. I went ahead and used a micropipette because I found that easier to mix the contents inside the microcentrifuge tube. Once you mix the Matrigel very well with the DMEMF12, you can go ahead and transfer that entire mixture into the rest of the DMEMF12 in the 50 ml conical tube. You can go ahead and discard the microcentrifuge tube and the pipette tip. Prior to plating the Matrigel mixture, go ahead and mix it very well. And we're going to plate this on 6 well cell culture plates. So you're going to want to add 1 ml of this mixture into each well. Shown in the video, but prior to moving on to the next plate, I recommend that you actually mix the Matrigel contents in the plate that you already plated. That will just ensure that the Matrigel is fully distributed among the entire well, or throughout the entire well. And then now you're just going to do the same process for the other plate. You're just going to add 1 ml of the Matrigel mixture. So now what you do, go ahead and mix them, and let that sit for 1 hour in the biosafety cabinet. So now what we're going to do, we're going to go ahead and thaw out our hiPSCs, and we're going to be retrieving those from the liquid nitrogen tank. So as you can see here, Dr. Zhao is taking out the container that contains the cells. She's letting the liquid nitrogen actually drain back into the tank. You don't want that liquid to fall onto the ground. You want it to go back into the tank. So now she'll take off the safety, and she will take the box that contains her cells, and she will take out 1 cryogenic vial. Don't forget to lock it back, because if you don't, these containers could actually fall and go into the liquid nitrogen tank, and it's incredibly hard to take these out. You would have to drain the entire tank. You very carefully put it back. to prevent any splashes, and then go ahead and close the liquid nitrogen tank. So now you want to work very quickly to thaw out these cells. So what you will do first is go ahead and vent the cryogenic vial in the biosafety cabinet. If it doesn't open, go ahead and spray the cryogenic vial with 70% ethanol and then try to vent it. That usually helps. And go ahead and thaw. Once the cells have thawed, you'll actually see just a little ice chunk inside. That is okay. Go ahead and leave that ice chunk in there because by the time you actually are taking out your cells, that chunk will be completely thawed out. Go ahead and wipe down the cryogenic vial and spray it with 70% ethanol prior to putting it in the biosafety cabinet. So this right here is teaser media. So this teaser media was previously thawed and I put it on ice. I'm going to put these plates to the side. So again, these plates contain the Matrigel. Go ahead and mix the teaser media well prior to adding it to your 50 ml conical tube. I'm going to go ahead and add 5 mils of teaser media into a 50 ml conical tube. And then what I'm going to do, I'm going to take 1 mil of teaser media and I'm going to add it directly into the cryogenic vial that contains our hiPSC cells. So you only want to add 1 mil of teaser media, and you want to add it slowly to your cells because you want to prevent a molarity shock. Be careful when you do this because again, that cryogenic vial could only contain about 2 mils of media. So it's about full, or 2 to 2.5 mils. So when you're taking the cells back up with that serological pipette, be sure that you don't completely insert the serological pipette without taking up some of the media, because if not, you're going to actually have it spill over the cryogenic vial. So again, just be careful when you do this. You want to collect all your cells, and then you're going to want to go ahead and slowly put them back into the 5 mils of the teaser media into that 50 ml conical tube. So I took up a little bit more media just so I could ensure that I washed out the entire inside of that cryogenic vial to collect all of the cells. And then I'm going to go ahead and collect it all up, and then I'll put it back into that 50 ml conical tube. I can close that up, and we're going to go ahead and centrifuge that so that we can collect the cells into a pellet. So I'm going to set the centrifuge to 1200 RPM for 4 minutes. Make sure that you find an empty vial with the exact same amount of liquid to balance out the tubes. Once it's done, go ahead and carefully take out the conical tube and be sure not to disrupt the pellet. Spray down the tube and then you can place that into the biosafety cabinet. So now we're going to want to vacuum up all the supernatant, so we're going to use a glass Pasteur pipette to do that. Before I vacuum up any of the supernatant, I want to go ahead and check for bubbles. So if there are any bubbles, I want to pop those first with the glass Pasteur pipette. This will allow for the bubbles not to actually disrupt your pellet once you've sucked up all the supernatant. So you want to tilt the conical tube to the side, and you don't want the glass Pasteur pipette to touch the pellet. So just to avoid that, go ahead and don't pass that line at the bottom of the conical tube. You can go ahead and cover your conical tube. So now what we're going to do, we're going to go ahead and resuspend the pellet with more teaser media. So you want to set up your glass serological pipette. Be sure that you're using glass serological pipettes for this experiment. You don't want to use plastic serological pipettes because plastic serological pipettes contain a toxin that's released into the cell culture, and that toxin is actually really bad for hiPSCs. So again, please be sure to use glass Pasteur pipettes. Add the media to the cell pellet and make sure that the cell pellet is completely dissolved inside the media. Centrifuge the cells at 1200 RPM for 4 minutes. Make sure again you balance the centrifuge. So I also thought out another vial of teaser media, just because we needed more to combine the teaser media with ROCK inhibitor. Go ahead and spray down that tube, and then you can put it inside the biosafety cabinet. So here on ice, I have my ROCK inhibitor. So my ROCK inhibitor, I had that in the minus 20 degrees Celsius freezer, and then I'm going to put it in the biosafety cabinet. I'm going to mix the teaser media with the ROCK inhibitor. The ROCK inhibitor is at a 1000x concentration. You're going to want to dilute that to a working concentration of 1x. teaser media to my empty conical tube. Be sure to mix the media before you take up media that you need. A micropipette to go ahead and add the ROCK inhibitor to the teaser media. So again, ROCK inhibitor is known to allow iPSCs to grow more efficiently. Be sure you use the correct micropipette tips. So now I'll add the ROCK inhibitor. This is a very small volume, so be sure that you're adding the correct amount. Use proper micropipetting techniques to do this. Because it is a small volume, go ahead and add it directly to the teaser media. Don't add it to the side of the tube, because if not, it might not mix well in the media. So add it directly on top or in the media. And then you're going to mix that prior to using it. I make a mark on the ROCK inhibitor every time I thaw it, just so that I know how many times I've thawed it. I'm also going to label the media because it looks exactly like teaser media. I want to make sure that this is a teaser media that has the ROCK inhibitor. So you only culture the iPSCs with a teaser plus ROCK inhibitor for 24 hours. After that, you're going to want to remove that media, and you're only going to be using teaser media to continue to culture the cells. We're going to go get our cells because they should be completely done. They should be pelleted at the very bottom. Again, do not disturb the pellet. Walk very slowly back to the BSC cabinet. Spray down the 50 ml conical tube. I'm going to vacuum up all of the matrigel mixture, because at this point the matrigel should have already settled on the cell culture dish, and then the rest of it is actually just excess. So again, we could go ahead and vacuum this all up. At this point, the matrigel has already been sitting in the cell culture dish for about an hour. So that's why we're able to completely take out the excess. Do not take out the excess before an hour, because if not, your iPSCs won't be able to attach properly onto the plate. Do the same thing for the other cell culture dish. is actually add our teaser media with rock inhibitor into each well. So you want to do this prior to adding your cells. Mix the media very well to ensure that the rock inhibitor is evenly distributed in the media. And you're going to want to add 1.5 milliliters of a teaser plus rock inhibitor media into each well. Once you've done that, you can go ahead and put the pipette gun to the side, because you could still use that same serological pipette to plate your cells. Again, be sure that there aren't any bubbles in the supernatant before you remove it, because you don't want those bubbles to be on top of your pellet. Again, you're tilting the conical tube and removing the supernatant very gently or very carefully in order to not disrupt the pellet. Now what you're going to do is you're going to resuspend the pellet in the teaser plus rock inhibitor media. Using your glass serological pipette, take up 6 mils of that media and resuspend your cell pellet. So we're using 6 mils because we already have 1.5 mils of that media in each well. So that means you're only going to be adding 0.5 milliliters of your cell solution into each well. And because you have 12 wells, that's why we're resuspending the pellet in 6 mils. Pellet, and very carefully you're going to want to resuspend it. At this point, I would recommend checking the speed of your pipette gun. You want to have the speed be very low to prevent any bubbles from forming when you're resuspending the pellet. If you form too many bubbles, you might not have enough cell solution to plate into each of the wells. I made the mistake here of not checking. So again, if you didn't check prior, please do check to prevent the formation of those bubbles. in order to plate the cells evenly into each well. Adding 0.5 milliliters of the cell solution into each well. I like to add drops into the well to prevent me from adding too much. You add it drop by drop. Because it's such a small amount, you could actually just count drops instead of adding instead of watching the pipette volume or the volume in the pipette keep going down. It's actually easier to count by drops. So now that we're done with that, you will incubate that, and now I'm going to go ahead and explain the feeder-dependent culture system. So to do that, you're going to want to go ahead and thaw out your cells. So again, here she is draining the liquid nitrogen back into the tank. Whenever you're taking cells out of the liquid nitrogen tank, make sure that you have proper PPE on. So Dr. Zhao has a face shield to prevent any of the liquid from splashing onto her face. Again, work quickly to get the rest of the contents back into the liquid nitrogen tank. Very slowly put everything back into the liquid nitrogen tank to prevent any splashing. You're going to take the hiPSCs and then you're going to go ahead and vent the tube inside of the BSC cabinet. And now you're going to thaw. So again, for every 1 milliliter inside of the cryogenic vial, you're going to want to thaw it out for 1 minute. So again, you want a little icicle about that big inside of the cryogenic vial. That should melt on its own once it's inside the biosafety cabinet. So here we already have a plate that has been incubated with MEF cells. So again, these MEF cells are going to serve as the feeder cells. So the hiPSCs are going to grow on top of the MEF cells. So Dr. Zhao is using a glass pasture pipette to take out all of the cells. So you could do this instead of what I previously showed. Either technique is fine. So you would add all of the cells into the 50 ml conical tube. Dr. Zhao is going to add hiPSC MEF complete media. The complete media is going to be added to the cells. Again, add it slowly to avoid that molarity shock. Adding it at about 1 ml at a time. cells at 1200 RPM for 4 minutes. These iPSCs were frozen down using collagenase. So collagenase actually keeps the iPSCs in clusters, and you don't want to break up those clusters. That's why for this experiment, we're not going to centrifuge twice like we did in the previous experiment. Because again, every time you centrifuge, that's actually, it could break up the cells, so we want to prevent that. So here we have the plate of MEF cells. So we're going to want to take out that media that we used to culture the MEF cells. We're going to want to rinse the MEF cells using media. So we use basal media in this case, so we use DMEMF12. Very well. Go ahead and remove that spent media. So here we use basal media, but you could also use PBS to rinse off the cells. So now what she's going to do is she's going to add iPSC MEF complete media into each well. She's adding 1 ml first to each well to ensure that the cells don't dry out. You could definitely do that first. So here I actually took out the cells from the centrifuge and I sprayed down the conical tube and gave it to her. So as you can see, the cell pellet is at the very bottom. You'll remove the spent media. All the cells are collected at the bottom. You're going to resuspend that cell pellet using the iPSC MEF complete media. Again, be very careful when you're resuspending the pellet. You're going to add the cells drop by drop into each well. Here, Dr. Zhao added cell solution 3 wells at a time, and she mixed in between each time she added the cells. Again, this all ensures even plating. Be sure to look at your cells underneath the microscope, and that's how they should look like. Go ahead and place them in the CO2 incubator. And remember to shake the plate forward and backward, side to side, inside the incubator to ensure even plating.